So part 3 of microbial genetics is going to focus on one more mechanism of how bacteria can change over time. And unlike mutations, which is common across all organisms that have to copy their DNA, this next is kind of unique to bacteria. And so what we want to talk about first is this whole principle of vertical gene transfer versus horizontal gene transfer. So these are obviously directions, and so you have to remember what they mean. So vertical is going to be up and down, and horizontal is going to be side to side. And so what we're talking about is, if we think about bacteria, they do have vertical gene transfer because during reproduction and binary fission, you have one bacteria generating two, and there is genetic material being transferred to each of those daughter cells. And so vertical gene transfer is going to be looking at kind of like the levels of a family tree. So with each level, you're at a different range of ancestors. So obviously you and your brothers and sisters and cousins are kind of on the same line, and then parents and aunts and uncles are above that, and then grandparents and so on and so forth. So in vertical gene transfer in humans, there is this process of genetic recombination because we use sexual reproduction, and during that process, you can get new combinations of genes during the formation of sperm and eggs, and so you get genetic recombination. But in bacteria, you don't get that genetic recombination because it's just asexual binary fission, so no new combinations of genes. But bacteria can do something else that we can't do, which is called horizontal gene transfer. And so horizontal would look something like this. So you have two bacteria, and this is they're not reproducing. This is just bacteria in the population. But there are mechanisms of DNA being transferred horizontally through one of these three mechanisms. So these are the three specific mechanisms of horizontal gene transfer. And it's this horizontal gene transfer in bacteria that can lead to this new sequence of DNA or recombination or genetic recombination. So mutation is a really important part of how bacteria can evolve with just random mutations that occur that could lead to a beneficial trait, but then that trait could spread through these horizontal gene transfer mechanisms over time. And so that's what we want to look at is these three types of horizontal gene transfer. One's called transformation, one's called transduction, and the other is called conjugation. So we're going to move through them in that order, starting with transformation first. And so what transformation is, it is the taking up or the acceptance of a bacterial cell of DNA from their environment. So this illustration kind of shows what's going on here. So if this bacteria dies, and bacteria die all the time from just a natural part of the cell cycle to, you can think of antibiotics killing bacteria. So as a bacteria dies, this blue represents pieces of the bacterial chromosome, or we can say pieces of DNA. Okay, so those are donor pieces of DNA. We don't know what information is housed on each of those pieces of DNA, but all DNA has some sort of instructions embedded within it based on the sequence. And so here is the recipient cell, and we call these cells competent cells because their competent cells are able and capable of being able to take up DNA from their environment. So not all bacteria are competent, so they can't all do this process, but some are competent, which means they can take up DNA from their environment. And so here's this process of transformation. There's a piece of donor DNA being taken up, so that's transformation. And not only is it taken up by this recipient cell, it can actually be incorporated into its chromosome and then used just like all the other parts of the chromosome. And so what I mean by that is whatever information and whatever trait happens to be stored on this piece of DNA, this bacteria now has it, and so it could get that trait as well. And so some traits that we're interested in are things like virulence genes. And so that's a term we're going to see in the future because virulence relates to the ability to cause disease. Another term we've seen is resistance genes. So that is about antibiotic resistance. And so those sort of traits are obviously really of interest in the field of medicine because it explains how virulence genes and resistance genes can spread through at least this mechanism of what's called transformation. So this is kind of the shortest summary that I could come up with. So a dead or lysed, so it's kind of exploded, donor bacterium releases DNA and fragments of its DNA into the environment. So those are those little blue bits. There are recipient bacteria present. Not all bacteria are competent. Again, competent means they are capable of taking up DNA from their environment. So if there's a competent recipient bacterium nearby, they can take up that donor DNA fragment, and that can be incorporated into their existing DNA, into their existing chromosome to create a new combination. And so again, that process is what we call recombination. And so transformation is one of the horizontal gene transfer mechanisms that can explain how we get new combinations of genes within bacteria or how we get recombination. So that's the first, called transformation. The second is called transduction. So it starts off with the similar beginning. So there's transformation and there's transduction. So it is one that students can frequently confuse if you don't study it carefully. But what's important about transduction is it's going to utilize a virus called a bacteriophage to carry out this process. So the picture over on the right is an illustration of how transduction is going to occur. So here is a bacteriophage, and what a bacteriophage is, as it's defined here, it's going to be a virus that infects bacteria. And the way that it's going to do that is it's going to physically interact with the bacterial cell, which is shown here kind of in this beige color. And then what's going to happen is the viral DNA is actually injected into this host cell. So remember what DNA can be. It's the instruction manual. So prior to this viral infection, the only instruction manual was the bacterial chromosome, shown here in purple. But now there is a new piece of DNA, and so you can think of this as a new set of instructions that weren't present before. And so these instructions are going to tell the cell to do something very different than what the original instructions told the cell to do. And so part of that is making some new enzymes that actually digest or destroy the chromosomal DNA. So here it's nice and intact, but now here it's all chewed up into pieces. So this is like shredding the bacterial instruction manual. And so now the only intact instruction manual is the viral DNA. So just notice the color coding. This teal color is viral nucleic acid or DNA, and the purple represents the bacterial chromosome fragments that are digested. So what this viral instruction is going to tell the cell to do is make a whole bunch of new viruses. And so that's what's happening here. Here's a newly made virus, and all of them have this nice teal blue color in it, indicating that they have made new viruses with viral genome in it. But this is the mistake that we're interested in for this process. So in this newly packaged viral particle, there isn't viral DNA, but it's purple in color, which would indicate it's actually bacterial chromosomal donor DNA, some fragment of it. So when this cell lyses or explodes and all of these are released, each one of these looks just like the normal process. So that's how the virus would continue to replicate in its replication cycle. But this is the phase we're interested in. It's called the transducing phase, because inside of it is bacterial DNA from the donor. And so when that finds a new host cell and it injects the DNA, it's not viral DNA that's entering, but again, it's bacterial chromosomal DNA from the donor. And so that's how we get horizontal movement or horizontal transfer from one bacteria to another. It's through this virus intermediary that happens to be carrying it and then injecting it. So similar to what we saw before, now that this new piece of DNA is present, it can integrate into this recipient cell chromosome. And so now we have this new piece of DNA that carries potentially a new trait of virulence or resistance or whatever the case may be. And so here's the summary of this. But notice what's most important is that there is a bacteriophage that's going to be carrying out this process. So there's a transducing phage that's carrying donor bacterial DNA. When that finds a recipient cell, it's going to inject that donor DNA into the recipient, and then that DNA, now that's in the recipient, can become incorporated into the existing DNA. So we now have two pieces of DNA that weren't together, but now they are. So we have that process of recombination through a horizontal gene transfer mechanism. So that's the second of three horizontal gene transfer mechanisms called transduction. And the last is conjugation. So we've actually talked about conjugation before because there's a structure that is used called a pilus, sometimes called a sex pilus or a conjugation pilus, and it allows two bacteria to connect. So this tube is a pilus, and it's linking one bacteria to the other. And this tube is like a tube is a good description because it's hollow, and so there can be movement of DNA through this pilus from one bacteria to the other. So in this case, there's two live bacteria, and they're going to be designated. One is F plus and one is F minus. And what that means, the F plus bacteria is always going to be the donor bacteria, so it's going to be giving DNA to the recipient. And the reason this F plus is the donor is it has something that the F minus lacks. So that's the reason for the plus. And what it has is a specific plasmid that's called a fertility plasmid. And so if you don't recall, what a plasmid is an extra piece of DNA outside of the chromosome. So we'll see this in just a second in the picture. But what you won't need to know now, that in conjugation, there's always two bacteria. There's an F plus and an F minus. And what makes the two different is the F plus is going to have the plasmid, which means the F plus doesn't have the plasmid. And so that's what we're going to look at. It turns out that there's two modifications of conjugation that we're going to go through, but everything that I've said so far applies to both forms of conjugation. You always have an F plus and an F minus. The F plus has the plasmid. The F minus doesn't have the plasmid. And it's the F plus that's going to be the donor, which means F minus is the recipient. Okay, so one example is just as I described. It's an F plus to an F minus. And so there's a series of steps that's going to happen. So remember what F plus means. It has the plasmid, so there it is. This is the chromosome, so that's there as well. And it's the F plus because the information that's on this, on the information that's on the F plasmid is what allows for the formation of this conjugation pilus. So an F minus can't initiate this because it doesn't have the plasmid, but because the F plus has the plasmid, it has the information on how to form a pilus and start the process of conjugation. Okay, so that's what we see in step 1. The F plus donor makes the pilus and draws the two bacteria close together. Then what's going to happen is we've already said the F plus is going to be the donor, and this is going to be the recipient. But what we want to note is which of these two DNA molecules is going to be transferred through this pilus. So again, there's that little connection or bridge called a pilus. And what gets transferred? One strand of the F plasmid, and so that's being shown right here. So this is a double-stranded piece of plasmid here, the fertility plasmid or F plasmid, and only one of the strands moves through the pilus. And so what you get, it doesn't necessarily show it, but you'd have a single strand here and a single strand here, because it started as double-stranded and one moved here. So you have single-stranded DNA here, and then you'd be left with single-stranded DNA here. But as we've seen before, in cells, there is the ability to take one strand and make a complementary copy of it. And so that's what happens from this kind of third panel to the fourth. The single strand becomes double-stranded DNA again. And this single strand becomes double-stranded DNA. And so now what you have is what was once the F minus cell that didn't have the plasmid, because that's the piece of DNA that was transferred through the pilus, you now have an F minus cell being converted to an F plus cell, because, as we said, what defines an F plus cell is it has the F plasmid, and this cell now does. So this becomes F plus, this stays F plus, but then realize what can now happen. Both of these cells have the fertility plasmid, and they can go and conjugate with another F minus cell. And so this can help explain how anything on this fertility plasmid could grow in the population, because it can be transferred from one donor to one recipient cell with each round of conjugation. Now, what I haven't said is what is on the fertility plasmid. So the only thing that we know for sure that is present on a fertility plasmid is all the information necessary to initiate and carry out this process of conjugation. So that's the bare minimum. That's why it's called a fertility plasmid, because it allows for this process of conjugation. But there can be additional genes that are present on the F plasmid that could be also important to understanding how bacteria evolve and change over time. So again, the two examples that I keep going back to is virulence genes. So if there's a virulence gene on this plasmid, then that's being transferred to this recipient cell, and now it has a new trait that could make it better able to cause disease, because that's what virulence means. Or it could be a resistance gene, or genes plural. So now this bacteria that wasn't resistant to a particular antibiotic may be resistant to one or more antibiotics after this conjugation event. So this is a really important mechanism of horizontal gene transfer, just like the other two that we previously talked about: transformation, taking up DNA from their environment, or transduction, where a bacteriophage is transferring bacterial donor DNA. There's one more type of conjugation that I want to talk to, and this is really a special type. So this is special F plus cell that is going to get a different designation called an HFR cell. So we still have conjugation, so you have an F plus and an F minus. The F plus cell is going to be the donor, and the F minus is going to be the recipient. But what makes this different is it's not going to be exactly like the F plus cell we saw before, because it's going to be a special type called an HFR cell. And if you look at the picture here, or you read this information here, what makes an HFR cell different from an F plus cell is plasmid DNA integrates into the chromosome. So prior to this event, they're separate DNA molecules. Here's the chromosome, here's the F plasmid, and they're separate, and that's how we defined an F plus cell. But to convert to an HFR cell, this plasmid integrates or becomes one with the chromosome. So rather than having two separate pieces of DNA, it's one continuous piece. So it's still classified as an F plus cell because we can say, check, it has the plasmid, but by using this designation HFR, we now know the plasmid isn't separate, but rather it's integrated into this bacterial chromosome. Okay, so because of that change, we're going to get a different outcome during the events of conjugation. Okay, so there's some similarities, but there's some differences. So the first difference is, where is the plasmid? Is it integrated or is it separate? And so that will determine which type of conjugation that's going to proceed. So in an HFR, specialized F plus cell, we're still going to get DNA being transferred, but notice it says part of plasmid, but really what's mostly transferred is donor DNA. So purple represents bacterial chromosome, and so there's a little bit of plasmid DNA at the leading front edge of what's being transferred, but the vast majority of what is transferred, as it says here, mostly donor chromosomal DNA, is sent through that pilus. And so that's what gets transferred over to the recipient cell. What that means is this cell, the recipient cell, does not get the full plasmid, just a part of it, and so it remains F minus. This one stays F plus, but this recipient cell has now a fairly large piece of donor DNA that could get incorporated or recombined into its chromosome, making a new combination of genes that didn't exist before. Again, how that changes the bacteria is unknown. It depends upon what was present before and what is present in this new piece of donor DNA that it received. But what it's showing is a mechanism that allows for the combination of DNA that wasn't combined previously. So in this example of HFR, which is a specialized F plus cell, you still have the F plus forming the pilus, you have the F plus donating DNA, but because of this specialized type of F plus cell where the plasmid isn't separate, it's integrated, it changes how and what DNA is going to be transferred, where it's mostly chromosomal, and so the recipient doesn't get the plasmid, the full sequence of the plasmid, so it remains F minus, but it does get new donor DNA that can lead to new traits that it acquires through that new piece of DNA. So this last slide is really just summarizing some of the basics of these three types of horizontal gene transfer. So this is the reverse order. This is what we covered first, which is transduction. You get some donor that's lysed. Actually, sorry, we did transformation first. I apologize. So some cell dies, and so there's some donor DNA floating around, and a competent recipient cell can take up that DNA to become transformed. In transduction, there is a donor cell that has been infected by a bacteriophage, and during the lysis event of that bacterial cell, there could be a phage that is called a transducing phage that's carrying donor bacterial DNA rather than viral DNA, and so that can lead to the transfer of bacterial DNA through transduction. And then what we just ended with, the third example, and again, there is kind of two different ways that conjugation can occur. There is the F plus or the HFR, specialized F plus, but both are going to involve the formation of a pilus and then the transfer of DNA from the F plus to the F minus. And so the recipient is going to receive new DNA that it didn't have previously. And so these three horizontal gene transfer mechanisms are really important for explaining how bacteria can acquire new traits, because they don't get recombination doesn't occur during vertical gene transfer like we see in sexually reproducing animals or sexually reproducing organisms, but instead what we see is this kind of odd way. I mean, it's odd from a human perspective because we can't transfer DNA in this way, but bacteria can. So it's a really important mechanism to explain how bacteria can change and evolve over time.